All right, so we're going to move into 7.7 of Unit 7, which is going to be Punnett squares. And it's going to be a little bit longer of a presentation, so feel free to split this up over a couple of days. Also going to have a lot of practice that goes with doing Punnett squares. I will try to post some supplemental videos for our virtual learners only at a later date so you can actually see the process some more. We may have to schedule a few Zoom meetings with some of you to work on this in a live setting. If you need that, be sure you send me an email or a message and let me know. So probability is the likelihood that something will occur. So with Punnett squares, we're basically figuring out the probability of a certain trait or allele to show up. Whether it's a monohybrid cross or a dihybrid cross, it's all going to be based on a percentage or a fraction. Law of segregation, this states that each allele has an equal chance to combine with the corresponding allele from the opposite sex. Punnett squares show how these probabilities can be demonstrated. Punnett squares are going to be diagrams that can determine the probability of genetic crosses. So alleles are going to be arranged on the top and the left side of the box, and you'll see what I'm talking about on the next slide. The individual squares of the Punnett square show how the probability of the offspring are inherited with a given trait. We're going to assign letters should be grouped together and arranged with a capital letter in front. I should say front, not from, and with a lowercase letter in the back. Genotypic and phenotypic percentage, each Punnett square again is going to be dealing with probabilities. You can write that as a fraction or a percentage. So if you look at a Punnett square, capital T, lowercase t, capital T, lowercase t, that is our genotype of the P generation, P1 generation. And so the boxes in here could represent our F1 generation. And you'll see that we take the genotype, a heterozygous genotype, and separate it, capital T on one box on one side and the lowercase t beneath it, and same thing at the top. And then what you do, let me get my pointer. What you do is you take the line from each of those, basically imaginary line, and you fill in the box in the middle. All right, so you have two capital T's, homozygous dominant. Here you're going to have heterozygous. We have a capital T, a lowercase t. Here is heterozygous, and then down here is going to be homozygous recessive. Normally, if you can set up the Punnett square properly, you can conduct the inside or fill in the boxes fairly easily. So monohybrid cross is going to be a cross between two organisms when only one trait is being considered. So for the example we just went through, let's say that capital T is the dominant allele for tall, and lowercase t is a recessive allele, which is shortness. Principle of independent assortment says that individual traits are inherited independent of one another. So for example, a short pea plant with yellow seeds passes its genes for height and seed color separately. You can be a short pea plant with yellow seeds. You can be a short pea plant with green seeds. You can be a short pea plant with, or you can be a tall pea plant with yellow seeds, and you can be a tall pea plant with green seeds. Meaning this, the allele for height has nothing to do with the allele for color. Okay, it's not a bundled package, so to speak. Dihybrid crosses are a cross between two organisms in which two traits are being considered. So for an example, a tall pea plant with green seeds is going to be crossed with a short pea plant with yellow seeds. Each organism's allele combinations are gathered and combined using a Punnett square, which can use as many as 16 squares, but not always. This is where you'll use the product rule. Okay, product rule. So this is going to take some practice, but with the monohybrid crosses, you would just go AB or AABB, but capital A, lowercase a, capital B, lowercase b, and you would think that's what you would do here. That is not it, because remember, we're tracking two different traits. So each of those traits have to be present within each of these boxes, right? So you're going to do the product rule, which is first to middle, first to last, second to middle, second to last. Or it could be first to third, first to fourth, second to third, second to fourth. Okay? First to third, first to fourth, second to third, second to fourth. That is the product rule. Right? So how will we fill this out if we're taking this genotype? Again, this is a genotype. It's heterozygous and heterozygous. It would be first to third. So capital A, capital B would go right here. First to fourth, capital A, lowercase b, go right here. Second to third, lowercase a, capital B, will go right here. Second to fourth, lowercase a, lowercase b, will go right here. Okay. Even though right here we had a lowercase a and a capital B, even though the lowercase a is in front of the capital letter, I'm going to keep it that way because the a's are in front of the b's in this particular genotype. And then we would do the same thing with this genotype for the top. And then when you fill it out, you're actually going to have 4 alleles per box. Okay, and we'll practice. I went way too far. We'll practice that some more. We'll post some of the videos of actually showing you this process. Incomplete dominance is when more than one allele is dominant, and when they are combined, neither allele completely expresses itself, resulting in an intermediate condition. For example, below, in a certain type of flowers, snapdragon flowers, we see that red and white are both homozygous dominant. Well, what happens if you cross-pollinate between these two when there's no recessive trait? Okay, which one wins out? Well, in this particular organism, neither one of them went out. They blend into a pink color. There are some organisms where one trait may be favored more than others, even though that they're both dominant. But we also see this idea here of incomplete dominance. Then you have codominance, is when more than one allele is dominant and they are combined and both alleles are expressed, resulting in both phenotypes being present. So if we look in chickens, black chickens and white chickens are both dominant, homozygous dominant. And when they are crossed, then you're going to get a checkered chicken. Okay, you'll get a chicken that's got both white and black on its feathers. Multiple allele is when one gene has more than 2 allele possibilities, usually resulting in a hierarchy of dominance. So this is what we talked about, where there may be a few dominant colors in a particular organism, but some are favored more than others. And these are just important vocab terms that you need to know. Polygenic traits is when one trait has more than one gene that codes for it, usually resulting in a blending of phenotypes. All right, so an example is human skin color. A lot of different shades of human skin color, and it's going to be portrayed in a lot of different ways. All right, so sex-linked genes. All right, these are genes that are only found on the X or Y chromosomes. This is where you may really have to slow the video down a little bit and go back and watch a few times. This is a very important this is a very important concept that is sometimes kind of difficult for students to grasp. So feel free to go back and watch this as many times as you need. And starts out with understanding what males and females have. Okay, males have an X and a Y chromosome, and females have 2 X chromosomes. That's important. The genes for maleness are found on the Y chromosome. So go ahead and put a note somewhere in your notes that the Y chromosome only codes for maleness. That's it. So when we're tracking sex-linked genes in a sex-linked cross, we're not going to put any superscript or exponent, as some of you would call it, on the Y chromosome, because the Y chromosome is only going to code for maleness. So for instance, if we are tracking color blindness, which is a recessive sex-linked gene, then we're not going to put the lowercase b on the Y as an exponent, because Y only codes for, again, for maleness, and color blindness is not a maleness trait, even though color blindness is more common in males than it is females. So there's many important genes that are located on the X chromosome. An organism cannot survive without an X chromosome. Recessive genes inherited on the X chromosome have a greater chance of expression in males because they only have one X. Okay? So if there's only one X chromosome in males, then whatever's on that X chromosome is what the male's getting. There's no chance for a heterozygous type situation. Whereas in females that have 2 X chromosomes, one of the chromosomes can carry the dominant gene, and the other chromosome, X chromosome, could carry the recessive gene, and the recessive trait be drowned out. So sex-linked disorders like colorblindness are more prominent in males because of males only having one X chromosome. Sex-linked crosses. So in order to perform a sex-linked cross, a monohybrid cross between XX and XY should be performed first. If you need to remember what a monohybrid cross is, please go back in this video. Next, the dominant and recessive alleles should be placed on the parents as superscripted numbers. So again, the lowercase b is going to be the colorblindness trait that we're using for an example here, and uppercase B is, or capital B is the normal trait. All right, so we see here lowercase b, and uppercase B. So remember, the Y chromosome doesn't carry the same genes, so we will never place a letter on it. There will never be an exponent on Y. And this is the hardest thing for students to remember, and it's just a habit thing. Okay, you get in a habit of putting superscripts on chromosomes, X and Y chromosomes, but there's not supposed to be one on Y because Y is just the male gene. Last, simply perform the cross with the superscript letters, making sure they never place a gene on Y. And genotypic and phenotypic percentages can be listed with male and female separately. So this is what a sex-linked cross will look like completed. Okay, from a little bit different example from the previous slide, but you see you will carry out basically two monohybrid crosses here, and you get the superscripts based off the superscripts that are coming with the X's and the Y's, and notice that there's nothing on the Y. And then when we write the phenotypic and genotypic percentages, half the females will be carriers. Okay, so you got two females, so one of the two could be a carrier, other half will be normal, so on and so forth. All right, that is the end of this presentation. There will be some work next week that will deal with practicing with monohybrid and dihybrid crosses. So there's a form that goes with this video. If you have a question, please shoot me an email, leave me a comment, and we will get together and talk about it. Thank you.